This video is going to help you get prepared for tomorrow's essay test. And remember, this is not a normal essay test. This is going to be a test where you're going to have to do molecular drawing. So you're going to have to draw and label any of the items from the 4 menus that we used in Chapter 2. So without further ado, let's get this party started. First thing that we learned about was water. And if you look over here, you'll notice that water is made up of an oxygen and 2 hydrogens, hence the formula H2O. All right, let me get the color right here that I need. All right, so as you can see here, this oxygen is a bigger atom than hydrogen, so it's going to hog the electrons. So this will make water is going to be partially negative and the hydrogen is going to be partially positive because the electrons are spending more time here than over here. So essentially what's happening in oxygen is it's gaining electrons. When you gain an electron, you become negative, and the hydrogens are losing an electron, so they become positive. So think of water as polar. It's got a negative side and it has a positive side. Remember, this is called polarity, and this is where all the cool features of water come from. Things such as cohesion and its cousin, adhesion. All right, let's move over here to this one. When we got into menu 2B, we began to talk about dehydration synthesis and hydrolysis. In other words, how are we going to build these macromolecules and how are we going to break them apart? So what we have over here is just the basic picture. Now we also drew this on that little that frame and comparison table that you got yesterday. All right, so what we have here in red, this is simply a monomer. Remember, mono means one, mer means part. So there's a monomer over here, and there's a monomer over here. Now this functional group here is called a hydroxyl, and remember, hydroxyl makes this part of the molecule polar. But when we do dehydration synthesis and hydrolysis, we're going to use the functional groups. So what we need to do in dehydration synthesis is take out the water. So we're going to take the we're going to take the H away from there, and we're going to take this OH right there. That's going to give us our water. And since oxygen can bond with 2 things at once, we have another bond spot right there from the hydrogen we took. And that's going to put these 2 monomers together. And this is going to make a dimer, which is 2 parts. Okay, so this is dehydration synthesis. Remember, this is an anabolic reaction. And it's also going to be endergonic because you're going to be storing energy in the new bonds that you made there. All right. Now just the opposite down here is hydrolysis. The word means using water to break apart. So here's the water molecule, and really what you're going to do is you're simply going to break it apart, and then you're going to put the functional groups back here from where they came from. So over here, this is your dimer. You're going to break this water apart. You're going to replace the 2 hydroxyl groups, and you're going to get your 2 monomers. So there's a monomer there and a monomer over here. Now remember, hydrolysis is what we call catabolic because the cat breaks, and this is an exergonic reaction because it's going to release energy. All right, here we go. All right, carbohydrates. We have 4 biomolecules: carbohydrates, lipids, proteins, and nucleic acids. And the first one that we went over were carbohydrates. And the monomer of a carbohydrate is a monosaccharide. And we draw these in really simple geometric shapes just to keep it simple. So whenever you see a hexagon, you need to recognize that this is glucose. And then whenever you see a pentagon, that's also going to be a sugar. And in this case, it's going to be fructose. Now, when we get to nucleic acids, you're going to see another 5 carbon sugar called ribose or deoxyribose. They're also going to be drawn as a pentagon. However, you need to recognize, is it fructose? That would be in like a regular carbohydrate, or is it going to be ribose or deoxyribose? And you're going to find those in a nucleic acid. So make sure that you understand the structure of a nucleotide so that you can tell that we're talking about a carbohydrate or a nucleic acid. All right, so you take 2 monomers or 2 monosaccharides and you hook them together, you're going to get a disaccharide. And this is also a dehydration synthesis reaction. So you're going to take an H, whoops, did it again. You're going to take your H off of the OH group over here. You're going to take the OH off of that. There's your water, and there's your 2 bond. So the disaccharide is actually this thing. And remember, both of those up here, those are simply a monosaccharide. Polysaccharides, 3 or more monosaccharides hooked together. And here's a really simple one. We have 1, 2, 3 glucose molecules hooked together. So we're on our way to becoming starch. Starch doesn't exactly have this formation, but our keep it simple version is going to be this. All right, lipids are the only one of the biomolecules that do not have a true monomer. However, fats and oils do come in this triglyceride formula. All right, the glyceride part refers to glycerol. Glycerol is a 5-carbon, make that a 3-carbon alcohol. Whoops, let's see if I can spell carbon right. And it's going to have OH groups over here. Fatty acids are going to contain a carboxyl group and a long carbon chain. And so basically a triglyceride looks like a capital letter E, where the backbone is glycerol and then the fingers coming off are the fatty acids. So triglyceride is where that name comes from. All right. All right, amino acids, these guys are the monomer of a protein, and proteins are arguably the most important of the biomolecules. An amino acid has 1, 2, 3, 4, 5 items over here. It starts with this alpha carbon in the middle, so that's what the purple thing is. And then you stick a boring hydrogen on top. And then to the left, you have the amino group, which was an NH2. This one acts like a base. And on the other side is a carboxyl group. That one acts as an acid. A carbon to a double bonded oxygen to another OH group. Now, some people get confused that this OH is a hydroxyl group, but it is not. You need to look and see what this is attached to. If the OH is attached to a carbon with a double bonded oxygen, carboxyl. So amino acid. Now the R group is the variable. This is what makes one amino acid different from another. Now we can hook amino acids together and they're going to be held together by a peptide bond. Notice that the peptide bond is between the carboxyl group of one amino acid and the amino group of another. This one here has 1, 2 amino acids hooked together. So this is called a dipeptide. And if you have 3 or more of these hooked together, you have a polypeptide or a protein. All right. Very important picture right here. So make sure you got that one memorized. We're going to be looking at this one over the year forever and ever. All right. Enzymes are the most important function of a protein. These are the chemicals that basically make all the chemical reactions occur in your body. And without enzymes, pretty much life wouldn't exist because you need these things to control the chemistry that keeps living things alive. Now this here is a very basic model of an enzyme. And the shape of the enzyme is really important. So you see the shape of the spot right here, the active site? This is where all the work happens. Now the active site is for substrates. Think of substrates as the reactions, think of a substrate as the reactant in a chemical reaction. So these substrates right here, A and B, they're going to fit in this active site. So you can see over here in this picture, A and B are in the enzyme. Now, when the reactants or the substrates A and B are in the enzyme, this is called the enzyme substrate complex. And it only happens for just a brief moment. And this is when, in this example, A and B are going to be turned into the new product, which is C, which is right here. And now this C will leave the active site, and this enzyme can go find more of these guys and do this over again. So this is that lock and key model. You've drawn this probably 2 times. You've made the vine. You did the foam models. You should have this one down pat. So we got to make sure that we know this one. All right. Now enzymes can be turned off and turned on 2 different ways. One of them is competitive inhibition. This is the competitive inhibitor. We've used a CI for short. And what it does is it fits into the active site. So now A and B will not fit in here, and that reaction is essentially turned off. Now another way to inhibit an enzyme action is to do allosteric inhibition, also known as non-competitive. This is your non-competitive inhibitor over here. And you'll notice that when the allosteric inhibitor or non-competitive inhibitor is in this allosteric site, the shape of the active site has changed, and now A and B will not fit. And once again, this reaction is effectively turned off. Now, if we want to turn these reactions on, we just need to remove the competitive inhibitor, or if we remove the non-competitive inhibitor, their active site goes back to its regular shape, and it's game on. All right, our last two drawings are going to be of a nucleotide and ATP. Now, nucleotides are the monomer of a nucleic acid, and you can argue that these are the most important biomolecule because nucleic acids have the ability to code for the proteins. So think of a nucleic acid as the directions for making a protein, and then the protein can handle all the life's chemistry. All right, now a nucleotide has 3 parts to it. A 5-carbon sugar that I've drawn here in green. Now, depending on what type of nucleotide it is, if it's a DNA nucleotide, this sugar will be deoxyribose. And if it's an RNA nucleotide, this sugar is going to be ribose. Now, all nucleotides have the same type of phosphate group, and they're also going to have a nitrogenous base. Now, nitrogenous bases come in a couple of different flavors. Number 1, they can have 2 rings like the ones I've drawn here, or they can have a single carbon ring, but they all contain nitrogen, hence the name nitrogenous base. All right, you'll link these together using dehydration synthesis to make your DNA or RNA strands, and we're going to learn about that when we get to our next chapter, which will be chapter 12. And then finally, we have this nucleic acid or nucleotide-like molecule called ATP. It stands for adenosine triphosphate. And it's basically an energy transfer molecule. All right, so if we are going to have an exergonic reaction, make energy, that energy is going to be captured by ATP, and then ATP is going to transfer that energy over to an endergonic reaction. Okay, now the most important thing that we want to know in ATP is that the energy is stored in these purple bonds right here. These are called the high energy bonds. And we're going to learn more about how this works in chapters 8 and 9, which we'll get to right around Thanksgiving. All right, make sure that you study these pictures. You watch this video a couple of times, you have all these pictures in your notes, and make sure that you do a good job studying tonight so you can kick some butt on tomorrow's Celebration Knowledge. So until the next video, we're going to catch you on that flip side.